For Chapter 8, Video 4, I'm going to diverge a bit from vaccines. We're going to talk about DNA fingerprinting. This technique is important for diagnostics, for forensics, for paternity cases, and more. DNA fingerprinting uses what's called restriction fragment length polymorphisms, or RFLPs, in order to look for similarity. If you look, we have 5 samples of DNA. They're going to have some sequence differences because they may be from different individuals. You mix them all with the same restriction enzyme to cut them into fragments and then run them on a gel. See over here that the DNA from the different samples have different restriction recognition sites, which are represented by the gold lines demarcating where the cut site is. When you subject them to the restriction digest and then run them out on a gel, you will get the results shown over here on the gel. We load the samples in the wells at the top of the gel, and then you subject it to current. DNA is going to start to migrate towards the positive end of the gel because it has an overall negative charge. The DNA fragments separate based on size. After running the gel for a while, the small fragments are found towards the bottom and the larger ones are towards the top. On each end of the gel, you place a ladder. Ladders are a supply that you can purchase from a scientific supplier. Ladders are DNA fragments of known size. You run them out on your gel with your digested samples so you can compare your digest to the known fragment sizes of the ladder. You can look for similarities. Here, if you look, you can see these samples are different from each other. One has a fragment here and here. 2 has a fragment here. 3 has these 2 fragments, which are different than everything else. And 4 is different and has these 3 fragments. 5, again, has only 1 fragment. So it doesn't look like it's cut by the enzyme at all. Each of these samples has a somewhat different DNA fingerprint. This is similar to what they do when they try to match DNA left at a crime scene to potential suspects. You're looking for similarities in the pattern so you can match people. The different patterns that you get after amplifying specific regions of DNA and then using specific restriction enzymes and examining the resulting DNA fragments are unique to individuals. You can compare the patterns that you get to your parents, for example, to show paternity and maternity, and also the uniqueness of the individual. You will share fragments with both your parents, but have your own unique pattern of fragments. This is the basics of DNA fingerprinting. The fragments of different lengths result in a unique pattern for different individuals. Hence the name restriction fragment length polymorphisms, RFLPs. Polymorphisms refers to the many differences. This next slide is looking at vectors. As mentioned in the previous video, vectors are replicating pieces of DNA that are outside the chromosome and can be introduced into cells. Vectors have some universal properties. They have to be small enough that we can do something with them in the lab. They need to be stable inside a cell. Once you introduce it in the cell hopefully keeps it and replicates it and uses the information, the DNA instructions in the vector. We need to have a recognizable marker so that we know which cells have taken up the vector and which have not. Because not all of the cells will take up the vector when you attempt to introduce it. Then, for most purposes, we have to have a vector that will allow us to what's called express the gene, meaning the gene actually gets used as instructions to make a protein. When a cell uses the DNA instructions to make a protein, that situation is called expression of the gene. Often we need the protein, not the DNA instructions for the experiment or the treatment. There are different kinds of vectors, including ones that are viral in nature. There are transposon vectors and modified yeast chromosome vectors that can take in huge amounts of DNA sequence and maintain it within a yeast cell. However, one of the most common types of vectors is plasmid. Plasmids are those circular pieces of DNA that bacteria share with each other under normal circumstances. Plasmids can have fertility factors as well as resistance genes or other useful genes under natural conditions. However, we can actually manipulate plasmids fairly easily in the laboratory. There are lots of different types of plasmid vectors available. You can purchase them from a supplier with different characteristics already built in. We can see here on the right-hand side that we have a circular piece of DNA representing a vector. Plasmids will have restriction enzyme sites within them in key locations. The recognizable marker in this case is an antibiotic resistance so that we can plate bacteria subjected to the transformation reaction with the plasmid on a media that has the antibiotic in it. Any cell that doesn't have the plasmid would, of course, be susceptible and die. However, the bacteria that took up the plasmid now have the instructions on how to resist that antibiotic, and they'll survive and multiply. You know that the survivors present as colonies on your medium have plasmid. You can see that in this fragment, we have done reverse transcriptase to take a messenger RNA, in this case for human growth hormone, and made cDNA for that messenger RNA. The cDNA is ligated into a plasmid. Remember, to make this efficient, the cDNA fragment ends were clipped with a restriction enzyme, and the vector was opened by cutting with the same restriction enzyme, so that there are sticky ends that are complementary on each. The cDNA fragment and the vector are glued together with ligase. And then you transform this engineered plasmid into a bacterial cell. Only the cells that picked up the vector will grow on the media plate with antibiotics down here. All the other bacteria that were in our transformation reaction mix that didn't pick up the plasmid won't be able to grow on a medium with the antibiotic. That's why the genetic marker gene, the antibiotic resistance gene in the plasmid, is important. Transformation reactions can be performed in the lab. While transformation was documented as a natural phenomenon, remember the classic Griffith experiment? We can perform some laboratory manipulations to make the cells pick up the DNA so they become transformed. Cells manipulated in the lab so that they will pick up DNA from the surroundings are called competent. We can use some chemical methods that make cells competent. This is a common and inexpensive way to make competent cells. No special equipment is necessary. However, there are other ways to get cells to pick up DNA. In this top panel of the figure, a method that is used is an electric field. It's called electroporation. Basically, electroporation zaps the cells with electricity. The cells have to be prepared in a specific way for this. Electroporation blows some holes into the cell membrane, and that allows the DNA present in the test tube environment to enter into the cell. Then the cell repairs itself with the new plasmid DNA inside. This creates a recombinant cell because it picked up the DNA from the environment and kept it. Protoplast fusion is another method that is used to create recombinants sometimes. It's another chemical process. During the procedure, chemicals remove the cell walls. You must keep the cells in isotonic solution so that they don't break. Then you use another chemical to make the cells fuse together. You allow the cells to recover and rebuild their cell walls. Cells that survive this process will be recombinants that have some genetic material from each of the original cells. Not all the genetic information from both cells will remain, only some. You will have to screen the cells to find the ones that have what you want. In addition to the chemical methods and electroporation, there are little guns that can be used to blast DNA into cells. Basically, you just shoot the DNA into the cell. Or you might inject DNA in a single cell with a little tiny syringe under a microscope. This method usually is used for larger, more rare cells, like maybe a fertilized egg. When scientists are trying to introduce genes to make a genetically modified organism, this could be used to more precisely control what enters the cell, a cell that's not easy to isolate. So, transformation, electroporation, protoplast fusion, gene guns, microinjection are all different ways of introducing DNA into a cell. This figure from your book is illustrating a process of genetic engineering that is used fairly commonly by researchers today. In the example that's shown here, we're looking at genes that came from a hummingbird. The research must be looking for a specific gene that does something interesting in hummingbirds, but you could substitute any organism of interest for the hummingbird. You want these genes to be put into E. coli. There will be lots of different fragments from the genome of a hummingbird, and you want them placed into a vector and then into E. coli. You will need to know which vectors end up with a fragment. If you look right here, you can see that you have a bunch of different types of fragments, and when you open up the plasmid with the restriction enzyme, many times the different fragments get incorporated into the plasmid. However, sometimes the plasmid closes right back up without taking in a fragment. How would you know if that had happened? After all, an E. coli with only the plasmid vector and no fragment from the hummingbird is not at all interesting to me. There is a way to know. Some plasmids come with a built-in screening mechanism for this situation. For this particular plasmid, a common screening method is shown. This method allows you to see easily which colonies have what is called the non-recombinant plasmid. The plasmid without an insert in it. You can tell the E. coli with the uninteresting, non-recombinant plasmid from the E. coli that have interesting, recombinant plasmids. Well, how does this work? Let's look up here. What I want to draw your attention to is two genes on this plasmid. One gene is the ampicillin resistance gene. This gene allows the bacteria to grow on media with ampicillin antibiotic in it. It makes the E. coli with the plasmid antibiotic resistant. When you plate the transformation mixture on the media with ampicillin, any E. coli bacteria that has taken in the plasmid will survive and multiply. There's another interesting gene on the plasmid. Look at this gene called LAC-Z. LAC-Z encodes an enzyme. The enzyme is normally part of the LAC operon, but that's not so important now. The LAC-Z gene has been inserted into this plasmid because the enzyme is able to turn a substrate blue. The substrate starts out colorless, but after the enzyme clips it, the substrate turns blue. If you look here, when we open up the plasmid with the restriction enzyme, the plasmid opens right there in the middle of the LAC-Z gene. If you insert something into the LAC-Z gene, like what has happened with these three plasmids, the enzyme is not going to work. If you disrupt the enzyme gene, you will not make a functional enzyme. The result is this. If you have plasmid only in E. coli plated on a medium with ampicillin and the enzyme substrate, you end up with blue colonies. If you insert a DNA fragment into the plasmid, you disrupt the LAC-Z gene, so you do not get functional enzyme. So you can't get blue colonies. The LAC-Z gene is not producing functional enzyme, therefore you're going to end up with white colonies on the media. For the plasmid that has no insert, the the LAC-Z gene is still working. If you put it on media with the substrate in it, these colonies of E. coli turn blue. Look down here at this drawing of a plate that resulted from a transformation using this mix of plasmids. Any colony that is not recombinant, that doesn't have an insert in the plasmid, is going to be blue. Those blue colonies are not interesting. You don't want to spend time looking at these because they're just the plasmid you started with, the one you probably bought from a supplier. However, any colony that is white, right, these white ones, they're interesting because the LAC-Z gene is not functioning, so they likely have a DNA fragment inserted into the plasmid. Now you can study these and figure out if they have the exact fragment of interest to you. The colonies of interest, the ones worthy of more study and research, are the ones that are white. This method is called the blue-white screen. Not surprisingly, not every single recombinant method uses this. Sometimes it's not needed. For the example down here, I tried to insert a gene for red fluorescent protein in an experiment that I performed during a training workshop some time ago. You can see that after my transformation reaction, I got a lot of colonies growing on my plate. When I put my plate on a UV light, you can actually see that some of the colonies are glowing red. Some are looking white. Well, it's pretty obvious which colonies have the plasmid that took in the gene for the red fluorescent protein because they're glowing red. I can easily pick those and stick them into culture and grow them up. Now over here, you see that I have a culture that's making lots of the red protein that I was interested in. You don't always need a blue-white screen because there are other ways to screen for inserts. Visuals like this colored product make it easy as well. But the blue-white screen is handy when you're trying to get an insert that isn't going to give you some sort of visual like this red fluorescent protein did. These are the basic tools and approaches for genetic engineering. There's lots of useful applications for these different methods. If you think only about the field of microbiology, we know that there are a lot of organisms that have never been grown in the lab. The estimate is that 99% of the different microbes out there in our biosphere, we have not grown in the lab. We can know a bit about them because of their DNA. We can amplify up DNA using PCR and then do DNA fingerprinting and see that they're unique. We can do next-gen sequencing to sequence large regions of their genome or the entire genome and realize again that they are unique. We can study what exists in a particular environment, like our own microbiome. We can study our mouths, our gut, the areas under our armpits, etc., and discover the various community members and differences from person to person. We now know that there are over 500 species of bacteria that live in our mouth. We have not grown all of them in the lab, but now we know they exist because we have their DNA sequences. In closing this section on biotechnology, I want to emphasize that there are a lot of applications for genetic engineering. Not only can we come up with therapies and vaccines. Your book has a lot of interesting things to say at the end of the chapter, and I encourage you to read through it, although we're not going to talk about them all. In general, you should realize that genetic engineering allows us to make synthetic proteins with bacteria, yeast, or eukaryotic cell lines that can be used for lots of different things, including therapeutics. This is a huge, booming area where new drugs are being developed. Protein therapies are huge right now. Genetic engineering has been used to produce different types of vaccines. One type that has not yet been put to widespread use is a DNA vaccine. For this technology, an individual would be injected with the DNA of the recombinant plasmid. The concept behind this type of approach is that once the DNA gets into the cell, it will travel to the nucleus and then the genes are transcribed. The mRNA will leave the nucleus and go into the cytoplasm where it would be translated. The human cells would synthesize the pathogen proteins encoded in the DNA, and that will stimulate immunity. mRNA vaccines also allow the human cells to synthesize the pathogen protein to stimulate immunity, but they save a step. No need to travel to the nucleus for transcription. If mRNA is used, there's only a need for translation of the mRNA in the cytoplasm of the cell. Another unexpected benefit to using mRNA is that it negates the conspiracy arguments that the vaccines could alter your DNA. If folks know the basics of the central dogma, they'd understand that our cells do not have genomes of RNA, and thus the mRNA does nothing to the genome. Genetic engineering has been responsible for the development of what is termed subunit vaccines, which are purified proteins. More of this in a later chapter. So, in addition to the current mRNA vaccines in use, such as the Pfizer and Moderna vaccines, there are modified viral vaccines, such as the Johnson & Johnson adenovirus vaccine against COVID-19. This too is created using the tools of genetic engineering. Other applications of genetic engineering are to introduce genes from plant pathogens into plants, which makes them resistant to infection with those pathogens. Genes that encode insect toxins can also be introduced. Those types of applications are referred to as our GMOs, genetically modified organisms, and they elicit different reactions from different people. We'll probably revisit this topic in the future. Genetic engineering produces drugs, industrial chemicals, vaccines, resistant crops, glowing animals for study, and more. We can't spend time talking about all these applications, nor all the controversies, but we will dedicate some discussion time to this. I hope you've enjoyed learning about some of the tools of biotechnology and how they impact our lives. See you in the next chapter.